Hello, everyone, and welcome to today's webinar, Emerging Solutions for Non-Invasive Cancer Monitoring. I'm Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by NuProbe. To learn more, please visit nuprobe.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Nick Gallerani, Scientific Affairs at NuProbe. Nick, you may now begin your presentation.
Great, thank you. Good morning, everyone, and thank you for joining today's session. And thank you to LabRoots also for organizing this event. My name is Nick Gallerani, and I work in Scientific Affairs at NuProbe. Today, what I'm going to be talking about is NuProbe and a technology that we've developed called blocker displacement amplification, or BDA, which we have begun to apply to non-invasive cancer monitoring research. So there are three main objectives for today's talk. One is to briefly go over the current state, advantages, and limitations of non-invasive liquid biopsies in oncology applications. The second goal is to introduce the BDA technology and highlight some of those applications that we've begun in non-invasive longitudinal monitoring. And then lastly, I want to go over the NuProbe ourselves and highlight some of the ways that these technologies can be accessed by working with NuProbe. Okay, so as you may be aware, the emergence of liquid biopsy has enabled many non-invasive testing options for cancer patients, including recurrence monitoring, non-invasive tumor profiling, and non-invasive therapy selection, as well as early cancer screening. Liquid biopsy can refer to a range of different types of tests, but for NuProbe, we're mainly developing next-generation sequencing-based tests for cell-free DNA analysis, as well as peripheral blood analysis in the case of heme cancers such as acute myeloid leukemia. And with test modalities like NGS, we can extract broad genetic information which may give the patient insights into things like disease prognosis, which drugs are likely to be effective, and which ones we might expect to see resistance to. Now, one of the central challenges in liquid biopsy is that the tumor-associated analytes themselves are quite rare. And in contexts such as monitoring or early detection, cell-free DNA that originates from the cancer may only constitute 0.01% or even less of the total fraction of cell-free DNA. And just to broaden the scope of this problem a little bit, for both solid tumor and heme malignancy monitoring applications, we see similar rarity problems. In MRD, we want to be able to detect signs of recurrence earlier so that we can also act earlier on the cancer. But if a patient is responding very well, the amounts of cancer-associated analytes are going to decline significantly to the point where they may be undetectable by most methods, even if they have trace remnants of disease or small drug resistance clones, which may eventually drive relapse. While there are certain error-correcting NGS methods that have been employed that are able to detect mutations at very low levels, these may be costly to implement, and they also may potentially show poor performance at single loci. So beyond just low concentration of analytes and cost sensitivity balance, a lot of different challenges exist in the liquid biopsy space before liquid biopsy's potential can really be fully realized. I think some of these can be considered biological in nature. Some of these include concordance between tissue and liquid samples, as well as detection of other important genetic biomarkers such as copy number variation. But there are also a lot of challenges on the clinical implementation side. Some of these include achieving that balance of sensitivity and cost that I talked about, standardizing pre-analytical variables, and then also what to do with all of the results, especially for some of these ultra-low frequency events. At the current stage that NuProbe is at, we're we're not really addressing all of these issues, but as a company that is interested in this space, I think it's important for everyone in this space to understand the bigger picture of what we're looking at. So, like I said, NuProbe is a fairly small company that can only focus on so many things. So today, what we really want to focus on is the issue of low concentration of analytes and achieving this cost sensitivity balance. And so I'll introduce this technology, which is based on our BDA technology called quantitative blocker displacement amplification. This was first featured in a 2021 publication in Nature Communications, and this is available through open access on Nature Communications website. Okay, so this technology, blocker displacement amplification, is a rare allele enrichment method, which enables us to selectively enrich mutant alleles while suppressing amplification of wild-type alleles during PCR. This enables us to sensitively detect mutations at single loci below 0.01% VAF. And this method is flexible and can be incorporated into a wide range of modalities, including qPCR and NGS from NuProbe's perspective. But we've also recently engaged in development of digital PCR assays. We recently announced a deal with Bio-Rad to incorporate BDA technology into digital PCR assays. So on the horizon, we can look out for new, highly sensitive digital PCR assays from Bio-Rad that contain the same technology. So the way that this technology works is through a competitive hybridization reaction between a non-extensible oligonucleotide called a blocker and an extensible primer. The blocker is designed to be complementary to a known DNA sequence, in this case, the wild-type template, while the primer is designed to have a region of overlap between the blocker and primer. And so basically this region of overlap creates competition between primer and blocker. When a variant is present within a 12 to 30 nucleotide region, which we call the amplification window, at the 3' end of the blocker, it creates a mismatch. This mismatch shifts the favorability of primer binding such that the primer now outcompetes the blocker for access to the binding site at that region of overlap. And over multiple cycles of PCR, we can achieve significant enrichment of variants while suppressing amplification of the known sequence. And so over many rounds of PCR, this can lead to over 1,000-fold magnification of those variants. Now, several of these methods for rare allele enrichment have been described in literature, but I think what differentiates the BDA amplification approach is really its scalability. So BDA enrichment can efficiently occur over a broader range of temperatures compared to some of these other methods that have been described, such as, in this case, LNA clamp PCR. This has enabled us to really scale up BDA assays to NGS approaches, allowing low-depth sequencing analysis of low VAF mutations. Now, in order to effectively scale up to these NGS levels, we have also we've made a modification of the core BDA technology and incorporated unique molecular identifiers into our BDA approach. And this is really what quantitative blocker displacement amplification is. It's BDAs plus UMIs to improve quantitation and correct for polymerase error that occurs during PCR. So experimentally, in order to demonstrate this benefit of reducing the wild-type read fraction, what we did first was we created a set of reference materials, which consisted of wild-type DNA that was mixed with 9 different synthetic DNA G blocks, each containing a different SNV or indel at roughly 1% VAF. Using a standard NGS approach without enrichment, 88% of the NGS reads were wild type, whereas with QBDA, only about 2% of the total reads were wild type. We did observe a small fraction of unexpected variants, likely introduced by polymerase errors, but because we had this UMI filtering and bioinformatics pipeline, we were able to effectively filter these false variants out. So we then constructed a 20-gene NGS panel capable of detecting over 450 SNVs and indels in hotspots related to acute myeloid leukemia. And again, using synthetic reference material, we compared QBDA to NGS with UMIs, but without this enrichment. And even at VAF levels below 0.01%, and in some cases well below 0.01%, we found that all the mutations were detected using the QBDA approach. And in terms of quantitation, over 80% of the mutations were within twofold of the VAF determined without enrichment, showing that although we are modifying the VAF percentage of the sample, we are able to accurately quantitate the VAF in our library. And importantly, because we are using this wild type suppressing technology, we are able to detect these mutations using only 4 million sequencing reads total, a significant reduction compared to deep sequencing approaches. So next, we applied the QBDA AML panel to a handful of retrospective clinical samples collected from 5 AML patients at time of diagnosis and also during complete remission. So using QBDA, we were able to identify a mutation in the NPM1 gene at 0.0052% VAF in patient 1. And this was missed by both flow cytometry and conventional NGS. This patient later passed away due to relapse of the disease. And in a second patient, QBDA as well as other NGS did not observe NPM1 mutations. In this case, actually, flow cytometry did report MRD positivity. NGS results at later time points during relapse confirmed the lack of NPM1 mutations, but did detect de novo mutations in KDM6A and PHF6, which we think suggests that this patient experienced clonal evolution, driving their relapse. So while QBDA did not detect these mutations, this likely is due to the makeup of the initial prototype panel rather than the technology itself. Our NPM1 results were consistently concordant with these other NGS methods, but we didn't have KDM6A or PHF6 on the panel. But I think, the fact that we're in at least one patient, we were able to observe this NPM1 mutation at 0.0052% VAF that was missed earlier and appeared later on conventional NGS. Suggests that this technology is very promising for monitoring of residual disease applications in acute myeloid leukemia. But I think the content itself of the panel may need to be expanded to function as a clinical MRD assay. And I do want to also be clear that all of these samples here were used retrospectively. Currently, all of our products are for research use only. So with that in mind, we are actually a project partner for an FNAH Biomarkers Consortium project to establish new methods for detecting disease in AML. We don't have anything specific to share beyond mentioning that we were working on this at this time, but we're hoping to have some updates in the coming months. Now, with that said, in the publication itself, we do actually demonstrate our ability to expand out and broaden our panel coverage. So in that paper, we built a pan-cancer panel that covered 61 genes and over 6,300 mutations from the COSMIC database. And this consisted of 360 amplicons split across 2 tubes. And in the paper, we test a variety of different clinical sample types that were all commercially obtained, including cell-free DNA, FFPE, fresh frozen tumor tissue, and also PBMC samples. And we show that we're compatible with all of these different types of samples. But what I wanted to highlight was really data that was kind of similar to what I showed earlier with the AML panel. So using a number of contrived samples that we prepared similar to what I showed earlier, we demonstrated our ability to detect and quantify mutations below 0.1% VAF, and in some cases 0.01% VAF, and showed good concordance with a standard NGS method with UMIs but without enrichment. Suggesting that we can really significantly expand the content of our panels while maintaining the performance and the benefits of BDA. Okay, so just to summarize everything that I've gone through today, quantitative blocker displacement amplification, or QBDA, enables highly multiplexed enrichment of rare sequences such as SNVs and indels, and detects and quantifies mutations below 0.01% VAF using both tissue or liquid samples. QBDA reduces the relative fraction of wild-type reads in NGS, which enables high sensitivity mutation sequencing with reduced sequencing requirements. This potentially translates to better cost efficiency as well as turnaround time. QBDA has been applied to clinical longitudinal blood samples from AML patients, and we think that this has great potential for longitudinal monitoring and other liquid biopsy applications. We were looking to further establish collaborations that may be able to further demonstrate some of these benefits, some potential benefits. Okay, so I wanted to next shift gears a little bit and talk about NuProbe, the company, as we are a startup and people may not be fully aware of how our company works and how you can actually work with NuProbe and access some of these technologies that I've been describing. So NuProbe is a developer of molecular technologies mainly focused in PCR and NGS for improved detection of nucleic acid biomarkers. So NuProbe has its origins in academia. We are a spin-out lab from using technologies from both Rice University and the Weiss Institute at Harvard. With these we've continued that spirit of technological innovation and greatly expanded upon the original technologies that our company started with, and developed a broader range of technologies designed to detect different types of nucleic acid biomarkers. So the one that I talked about today, blocker displacement amplification, is designed for SNVs and indels. But we are also working on other types of biomarkers such as gene expression, gene fusions, as well as copy number variation. And so I wasn't really planning to go over these other technologies, but just briefly, in case these are of interest, we have had some other recent publications also available in Nature Communications highlighting some of these other technologies. Briefly, the first one, quantitative amplicon sequencing. This is an amplicon-based NGS workflow for ultrasensitive detection of copy number variation down to 5% single copy change in HER2, as well as mutations down to 0.1% VAF using low sample input. We also have expertise in primer design. We recently published another paper about one of our primer design algorithms for design of highly multiplex PCR primer sets that minimize primer dimer formation. This allows us to efficiently design new multiplex PCR and NGS assays. Okay, so we have this broad range of technologies, but how do people work with NuProbe? So what we mainly do is we leverage this portfolio of technologies and collaborate with companies in the biopharmaceutical, life science, and diagnostic spaces, and also work with researchers who may be able to utilize these technologies. The main things that we do are sample testing using defined NGS and PCR assays, And we also provide custom assay development services to support translational research programs. We are aware that, research, each individual case is often different and requirements may differ for different assays. And so we are prepared to rapidly develop new solutions for different biomarkers which you may need. Just to briefly cover the current offering for sample testing. So we have 3 assays that utilize the quantitative blocker displacement amplification technology that I described today. The first being the AML MRD assay, which I showed data for earlier, and then also the pan-cancer NGS assay. We also have an assay designed for melanoma targets. On the QA-seq side of things, we have a HER2 CNV assay, and then we also have solutions using RNA for gene expression, as well as a number of different qPCR assays. And all of these assays in a research use only format are available immediately for testing. I do want to describe how our custom assay development program works. So we follow a consultative model where our team meets with your team and discusses the specific problem that your team may be having with certain biomarker detection. And we come up with a solution that utilizes our technologies, our family of technologies. Generally, we can deliver a custom NGS assay of about 200 amplicons in about 14 weeks. And this is generally targeted at translational research programs. But if this is of interest, feel free to reach out. Okay, so that brings me to the end of this talk. So non-invasive liquid biopsies are an important and emerging field in oncology testing. NuProbe is developing a wide range of technologies which may have features that improve liquid biopsies, such as that quantitative blocker displacement amplification technology, which I described. And these technologies are currently available to biopharma, life science, and research through our sample testing and custom assay development services. All right, with that, I just want to thank everyone for joining, and we can take any questions.
Great. Thank you so much, Nick, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. Now let's get started. Our first question is, how can you quantify the variant allele frequency in the original sample after BDA?
Yeah. So BDA can accurately quantify the variant allele frequency of the original sample, but you're correct that BDA also alters the variant allele frequency that we're measuring in the library. So this calculation is automatically performed by our analysis software by taking into account the input DNA amount and UMI barcode and conversion yield. And to accurately estimate input DNA, we also include internal positive controls on the NGS panels that don't have blockers that enable us to quantitate molecules at several different loci in housekeeping genes. I also want to point out, since bioinformatics, that can sometimes be a bit of a black box. I think the most powerful piece of data that we showed in this webinar is that we highlighted some data from the publication where we did actually compare the quantitation accuracy of quantitative blocker displacement amplification against UMI NGS without enrichment, and showed good concordance between those two methods, showing that our method, in spite of altering the VAF during enrichment, we are able to quantify the original VAF of the sample.
Thank you. Another question here. Has the BDA technology been applied to other biofluids, specifically a CSF?
No, not at this time. That is something that we would definitely be interested in trying, because we think that the same benefits that we see in like blood or cell-free DNA should be applicable to CSF as well, but we just haven't been able to actually try that. That would be an area of interest for collaboration for us. Yeah, something we're very interested in.
Great, thank you. The next question, is BDA available as a kit?
Yeah, so currently we don't offer any kitted products. Instead, we offer RUO sample testing services at our Houston R&D facility, and that uses that menu of predefined NGS assays and other qPCR assays as well. Yeah, and like I also mentioned, another channel that BDA is accessible or will be accessible will be through Biorad and their digital PCR assays.
Great, thank you again, Nick, for your time today and your important research. We'd also like to thank LabRoots and our sponsor, NuProbe, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their questions. Questions we do not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.